Hello, this is Miss Dagenford again, and we do need to do a part 2 to our video, and that is going to continue our discussion about the cell membrane, where we left off was that the cell membrane is selectively permeable. And what we want to tackle is that this affects what things can go in and out of the cell. Now, I had given you some examples of molecules. We have sodium and chloride and potassium and water, oxygen, carbon dioxide, and carbon monoxide. Now that's not a complete list, but notice, think about what has to happen. If you're an animal cell, so if you're an animal cell, so let's think about that amoeba that we drew in our lab. So this amoeba is going to, just like us, need oxygen. So oxygen needs to go into the cell, and then we want carbon dioxide to leave the cell. That's a waste product. Now, if you're a plant cell, it gets a little bit more complicated because a plant cell, remember, has both mitochondria and chloroplasts. However, let's just take a look at a cell that is photosynthesizing quite a bit. And because it's photosynthesizing, it has those chloroplasts around that big central vacuole. And those chloroplasts are going to be consuming, we want carbon dioxide, and then they're going to give off oxygen. So think about the needs of the plant. You know, what's the cell? What are the cells needing to get in and out? And cells need electrolytes like sodium and potassium and chloride, so they need to go in and out in some manner. And then there's also glucose. We already know that our body uses glucose as energy. Um, and we already talked about muscle cells using a lot of mitochondria. And mitochondria are the ones that need glucose and are going to convert it to that ATP. So glucose is going to go into muscle cells and provide energy. So we're getting this idea that molecules need to go in and out, but I left you with this idea that polar molecules can't pass through, nonpolar molecules can. So that kind of gives this big question: how in the world do molecules get in and out? Now, there's a couple of things that we're going to look at. We're going to look at passive transport. Now, passive is a word that you've probably heard with in regards to people, like that person is very passive, or somebody is a pacifist. And passive means peaceful or not a lot of energy. You're a passive person. Well, anything goes. You're okay. Would you like to go to see a movie, or would you like to go to the Renaissance Festival? I'm okay. I could do either. You're passive. Um, well, passive transport in this case is that we're not adding any energy. There's no energy required for things to get in and out of the cell. Now, that doesn't mean there's no energy completely. Molecules have inherent energy to them. They have energy because they are molecules that are moving. And that's part of that kinetic energy that they have, the electrons that are moving and the energy that with which they are moving about in the molecule. So what we want to first establish is that diffusion is the movement of molecules from an area of high concentration to, oops, forgot to finish that, to an area of low concentration. Now, you encounter diffusion all the time. If you put sugar in your hot tea or hot coffee, you may dump the sugar into the bottom of your tea. And even if you didn't stir it, you would realize that eventually that tea or coffee becomes sweet. And you have this experiential knowledge that that sugar, even though it started out at the bottom of the hot tea, it's now spread. And of course, stirring it makes it go faster. But instead of sugar crystals, we're going to see it nice and evenly spread out. If you have a bottle of perfume and you take that bottle of perfume and you spray, now spraying is of course kind of introducing another idea. But let's say you spray it and you put it in one side of the room. Well, if I sprayed perfume on one side of the room and that's it, like I don't put a fan on, I don't, you would start to, if you're a person over on the other side of the room, um, it's an odd shape, man. You would start to see these you would start to smell that perfume from across the room. And what we're talking about is this idea that these molecules are spreading. They're going from an area that is a high concentration to an area that is a low concentration. Now this happens, let's go ahead and draw a model of this. It happens because if you have a high concentration of these molecules, and they are moving. And that's a pretty fundamental idea of these molecules. They are moving. And as they're moving, they're colliding with each other. So this one's moving that way, this one's moving this way, and this way, and that way, and all sorts of possibilities. And as they are moving, you can imagine like a pool table, the balls, or as they're knocking against each other, they bounce in, and as they bounce, they collide and then they spread out. So what was a high concentration of molecules over time becomes a low concentration. They it may be the same number of molecules, but they're spread out. And that's the idea of diffusion. We see and we encounter it all day long. I mean, the antiperspirant and deodorant and cologne and perfume and soap uh, business is dependent upon this idea that molecules are moving and spreading. You spray perfume on you and you do not want the perfume to stay on you, because if it stayed on you, nobody would smell it. So you're walking down the hallway and somebody stops you and says, wow, you smell really nice. And you're like, oh, I just bought this perfume, or cologne, or inevitably, you know, we have that, person that maybe sprays too much, and you can almost see the cloud of perfume around them. And that's diffusion. And again, we see it. We walk into the dining hall on pizza day and, oh, I know it's pizza day. I smell it. Well, we smell it because the molecules have moved. Now, the in the membrane, if let's go ahead and draw our membrane. And again, we'll just draw 10 of these phospholipid molecules. And let's not forget our nonpolar tails. Now the tendency is just to start to scribble, and I know that's the tendency. Just you cannot forget that these are nonpolar tails. Now, if these are nonpolar molecules, so let's take the example of the oxygen. So my blue dots here are going to represent oxygen, and this is going to be outside the cell. And again, dot, 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 that's just showing the membrane is continuing. If this is a high concentration of oxygen outside, and there's just a lower concentration, and the concentration again refers to the number of molecules in a given area or a volume. So if this is a lower concentration, we should see lower amount. So this is going to be a high concentration outside. This is going to be a low concentration on the inside of the cell. And so because oxygen is small and because it is very nonpolar, it can pass through very easily. So we see these oxygen molecules just zip through very easily. Now, there are going to be some that zip out on the other side, just because, again, these molecules are moving, they're colliding. But because there are more colliding on the one side with each other and passing through the membrane, what books often then show is just this large arrow, It's just a very common designation. A large arrow coming in and then a small arrow going out, which kind of indicates the net flow, the net direction of the molecules. Yes, there might be some oxygen molecules that move out, but overwhelmingly the molecules are moving inside because there are more of, they're moving inside because there are more outside. Now, again, this is going to be easy for small, non-polar molecules. They can pass right in between the nonpolar tails. However, the bigger you are, even if you're nonpolar, the bigger you are, the slower it's going to take, the longer it's going to take. It's slow. And the more polar and then the more charged, it's going to be very difficult. They're not going to be able to get through, if at all, very slowly. Now, in order to accomplish this, because let's face it, if you have something that, for instance, if you have glucose, and glucose is relatively bigger, it's a big molecule, but more importantly, it's polar, very polar. It's got all of those oxygen and hydrogens that have charges, and that's not going to mix with those nonpolar tails. However, we want glucose in. So let's real quickly just diagram. If you are this amoeba and you want to have the carbohydrates for energy, you want glucose, these little monosaccharides, you want glucose inside the cell. But if they can't pass through, you've got a big problem. Same thing with muscle cells, same thing with neurons. If you use glucose for energy, but if they can't get through that membrane, what do we do? How are these cells well designed for this purpose? Well, if we just sneak over here, and again, you'll get this picture to kind of practice. Some molecules that are unable to pass through, and just as a good example, that's glucose. What we see is that cells have proteins, and these proteins are going to be transport proteins. And by the way, remember the example of like the teddy bear factory from the Rough-ER and the Golgi. This is an example of one of those proteins. So if we have this glucose molecule, but notice this is a high concentration on the outside and a low concentration on the inside, glucose is going to diffuse through. Now what we need is a doorway to allow it to go through. And these transport proteins, sometimes they're called carrier proteins, as the glucose diffuses through, they're going to go through the protein, not the phospholipid tails. And now that does mean that the inside of the protein better be polar, or else we're going to have the same problem. So inside this protein, you would see these polar amino acids. On the outside, it needs to mix with the nonpolar tails. It's just a very nice arrangement of molecules. And so that's one way that allows things to get through the cell. And that's called facilitated diffusion. Now, facilitated, some of you in Spanish class will learn that facile means easy. Or if somebody facilitates something, that you are making it easier for them. You are facilitating that process. You are making it easier. You're helping them. All right, well, we're going to practice this on the next day in class. Have a lovely weekend.